Okay, now today we will see the types of targeted drug delivery system or the carrier used for drug delivery. First is nanotubes. They are hollow cylinder made up of carbon atoms which can be filled and sealed for potential drug delivery. They are called as nanotubes and they are of cellular scale and needle type, attaching the drug molecules to cancer as electrode in thermocells. See the figure of carbon nanotubes. Okay. Now nanowires are also used as a carrier. The nanowire are the pinpoint damage from injury and stroke, localize the causes of seizures, and detects the presence of tumor and brain abnormality. This type of nanowires will use to identify or to localize the causes of the seizures and also detect the presence of tumors and brain abnormalities can be detected, right? So it is technique used for potential treatment of Parkinson's and other CNS disorders. Okay. Now next nano carrier which is used in the targeting is nanoshells. So what are nanoshells? So they are hollow silica spheres. See the figure. Centrally they are the silica spheres which is covered with gold. Then it is called as nanoshells. The scientists can attach the antibodies to the surface, okay, and which enables the cell to target certain cells such as cancerous cell. Now application, so it is used for the technique, it has potentials for targeting cancerous drugs. Now next is quantum dots. The carrier used is quantum dots. Okay. Now quantum dots are minuscule semiconductor particles, right? Very small size, but particle has property of semiconductor particles that can serve as a sign or post of certain types of cell or molecules in the body. Now it also has capacity of targeting, or it has potential of targeting to cancerous cell. Okay, they are called quantum dots. Nanopores, they are again engineered into particles. They are hollow particles. They are so tiny that DNA molecules can pass through them, one strand at a time, and which allow high, precise, and efficient drug sequencing. Right. So it is very important for genetic engineering and biotechnology. Most frequently and most widely used carrier for targeting of genetic engineering and biotechnology is nanopores. Okay. Now gold nanoshells. The scientists use these gold particles or gold nanoparticles to develop ultra-sensitive detection system of DNA and protein markers associated with many forms of cancer, including breast, prostate cancer, in cancer treatment and genetic engineering. Okay, so it forms this type of carriers. Okay, then use of this gold nano carriers, it is again used for cancerous treatment and genetic engineering. The dendrimers, what are dendrimers? So they are precisely defined as they are the synthetic nanoparticles. Basically what they are synthetic nanoparticles having size range of approximately near to 510 nanometers in diameter. They are made up of layers of polymer surrounding a control core. In the central we are having a control core which is surrounded by the dendrimers or polymer layer of the polymer. The dendrimer surface contains many different sites and on which the drug can be attached or drug may be attached. Alright. So in gene transfection and medical imaging, the dendrimers can be used. Now in case of liposomes, as we know that liposomes are the vesicles in which aqueous volume is composed of membrane, lipid membrane, various amphiphilic molecules are used to form liposomes, right? So drug molecules can be encapsulated in aqueous space as well as into bilayer lipid layers, right? So the extent of location, matlab which how much amount or where the drug will localized, it depends upon physicochemical characteristics of the drug and lipids, right? If drug is hydrophilic, then it localized into center. If drug is lipophilic, then it is localized in the lipidic bilayer. Okay. Now next is neosomes. So the difference here is they are non-ionic surfactant vesicles. Vesicle is made up of non-ionic vesicles or surfactants, right? And it also can entrap both lipophilic as well as hydrophilic drug, either in aqueous phase or in the lipid phase. Okay. And it is reported to attain better stability than liposomes. Yes, it is having more stability and it is more useful for targeting the drug for cancer, parasitic, viral and other microbial disease, right? See the structure of neosomes. Instead of lipid bilayer, the lipid layer, this bilayer is made up of non-ionic surfactant, which is having both the hands, polar head as well as non-polar head. There is no use of lipids are there at the neosomes, right? So inside structure is hydrophilic drug, hydrophilic structure. So it is your aqueous drug can be loaded in the aqueous region of the encapsulated carrier, and hydrophobic drug is localized in the lamella, right? So this is neosomes, liposome and neosome. And the last one is ophasomes. So they are bilayer structure formed by using single chain unsaturated fatty acids, right? So it is also called as pharmacosomes, and it comprises of two main parts of pharmacon, matlab active principle, and cosomes, matlab carriers, and that has the drug assembled to form vesicles, right? So these are called as pharmacosomes. Drug covalently bound to lipid may exist in the coloidal dispersion as ultra fine macellary hexagonal aggregates. So then it is called as ophasomes or pharmacosomes. Virosomes, they are immunomodulating liposomes, consist of surface glycoproteins of virus, influenza virus, and immune stimulated reconstituted influenza virosomes. Muramyl dipeptide virosome must be targeted oriented, and the physiogenic characteristics could be exploited in genome grafting and cellular microinjection. See the structure of virosomes: lipid bilayer in central virosome cavity, and on the surface there is presence of glycoproteins, and therapeutic drugs are encapsulated in the surface in the center. Okay, drug molecules in the center, complete virosomes. When it attached to the cell membrane, then it release the fuse with this cell membrane. See the binding of virosomes to the cell membrane is by cell surface receptor. See this, right? And it will open the cell membrane and transfer its drug molecules into the cell membrane. Okay, it is called interaction of virosomes with receptor and the release of the drug into the target cells. Okay, now last one is cubosomes and nanocrystals. Again, cubosomes are liquid crystalline phase which form small cubic particles suitable for injection. Nanocrystals, though they are in a nanospud material with at least one dimension less than 100 nanometer and it's single crystalline. Okay, so more properly, any material with a dimension of less than 1 micrometer, 1000 nanometer should be referred as nanoparticle, not a nanocrystal. For example, any particle which exhibits regions of crystallinity, that is termed as a nanocluster based on dimension. Nanobots, nanorobotics is a technology for creating machines or robots at close to the microscopic scale with 10 to minus 9 meter. So nanorobotic refers to the still largely hypothetically engineered discipline which builds nanorobots in the size range of 0.2 to 10 micrometer and constructed in the form of nanoscales. Transferosomes, they are again carrier for target drug delivery. It describes lipid droplets of such a deformability that permits in easy penetration through a pores much smaller than the droplet size. And this transferosomes is a supramolecular entity that can pass through permeability barriers and thereby transport the material from the other side. They are more elastic than standard liposomes. Okay, so thank you. Here we have completed different carriers for targeted drug delivery. Carriers used for targeted drug delivery. Okay.